and metabolic syndrome is a cluster of interconnected metabolic factors, including insulin resistance, obesity, hypertension, and atherosclerosis. So, if I talk about the components of METIS which lead to cardiovascular disease, we normally have obesity, inflammation, adipocytokinesis, and insulin resistance. So, where mitochondrial dysfunction has seen to be one of a major key factor leading to CVD. So these are few mechanisms which lead to mitochondrial dysfunction in CVD, where I can talk about the role of ROS, along which leads to increase in ROS, which leads to decreased mitochondrial biogenesis, thereby leading to mitochondrial dysfunction. So in one of the preliminary data of our lab, what we found was IL-4 as one of an inflammatory cytokine led to increase in ADMA levels, which thereby leads to one of a major factor leading to CVD. So how come? So hypothesis of our study was that IL-4 along with ADMA, the ones which are involved in arginine metabolism, takes in account the role of hypoxic response, major factor being HIF1 alpha. And one of a downstream target of HIF1 alpha, which is already reported in literature, is MIR210. So the IL-4 ADMA along with HIF1 alpha, they all lead to mitochondrial dysfunction leading to CVD. So this is a few preliminary work which was done. So what we had done for this piece of data was we had prepared a METIS mice model. So we checked the high fat and high fructose diets, and along with the CHO diet model was being taken. So we checked all the components, basic components like the fat mass, blood pressure, cholesterol, triglyceride levels, along with the IL-4 and ADMA inos levels, which were being checked in the pulmonary features were there. But we have not yet checked what is the role which is involved in aorta, which is one of an obligate aerobic organ. So these are few findings which we had come up with, where we first initially checked the levels of IL-4, followed by ADMA. So we found was that in both the high fat and high fructose diet, the levels of IL-4 and ADMA were upregulated, thereby leading to increase in INOS levels, as can be checked through both Western blot and IHC. This led to increase in the levels of hypoxic response, that is HIF1 alpha, where we found that the levels of HIF1 alpha were more in HFA and HFR, thereby leading to increase in the levels of the downstream target MIR210. So there we also wanted to check what effect does it have on the mitochondrial turnover proteins like PGC1 alpha and TFAM. So these two are known to be the major transcription factors which are involved in mitochondrial genesis. So through both Western blot and IHC, again we checked that yes, in the both high fat and high fructose diet, the levels of PGC1 alpha and TFAM, they were found to be down regulated, thereby leading to changes in the mitochondrial activity. So for which we check the levels of complex 1, complex 4 activities along with the inner mitochondrial membrane potential. So for this, what we had taken up was the JC1 dye uptake that was being estimated. So there we found that the levels of complex 1, complex 4 and the mitochondrial membrane potential were found to be down regulated, thereby leading to leakage of cytochrome C from the cytosol. Thereby, we could see that there were increase in the levels of cytochrome C in both the HFA and HFR mice model. So thereby, we came to the conclusion that yes, when there is an increase in IL-4 and ADMA, then they might lead to increase in the hypoxic response, that's HIF1 alpha, leading to decrease in the mitochondrial biogenesis proteins like PGC1 and TFAM. Along with that, we also checked the levels of apoptotic marker, that is caspase 9 and cytochrome C, where we found they were upregulated, thereby all these factors culminating into the mitochondrial dysfunction, which is thereby characterizing what is known as cardiovascular disease. So this is one piece of work which we had done. So these are few, this part has already been communicated, and these are few future prospects where we'll be using Entagomir 210 along with L-arginine and HIF1 alpha siRNA so as to confirm the role of oxonitrative hypoxia-driven MIR210 causing mitochondrial dysfunction. We'll also use the exogenous mitochondrial donation by MSCs using MIRO1 so as to check the mitochondrial function restoration. And thereby now what we are working on the diagnostic part and maybe the translation part we can say is where we are profiling the inflammation and acute kidney injury biomarkers using real-time PCR, metabolomics, genomics and proteomics in both heart failure and STEMI patients. So here we have uptaken heart failure patients where they've been characterized into reduced, mildly reduced and the preserved ejection fraction as per the levels of ejection fraction. And STEMI patients are being uptaken where we have subdivided them under the older and the younger STEMI patients. So this is the workflow which I think you all can see that we'll be recruiting the patients along with few healthy controls. We'll be going ahead with the proteomics and the metabolomics and the lipidomics part. If we come up with a novel biomarker, maybe an early prognostic biomarker, then we may validate those particular specific biomarkers using various molecular assays like ELISA, or flow cytometry, or maybe the O-link one. So these are few markers which we have digged into the literature, and we'll be running both untargeted and targeted proteomics here. So this would serve a new maybe a kind of a data which will help us validate the role of already existing anti-inflammatory drugs which are currently in clinical trials. So these are just the introduction slides where we have talked about the heart failure. These are few papers which are already published where the role of LPA and apolipoproteins is already known, but not much is known about the inflammation part. So if I talk about why there is a rising cardiovascular events in young adults, so we all know it is because of the lifestyle behaviors and the widespread medication use like statins. So we can see that yes, there is a high prevalence of metabolic conditions in youth, wherein type 1 diabetes is found to be there in more than 18,000 young individuals affected. So if I talk about AKI, it is being seen that mainly the patients who are at transplant or the ones who are at dialysis. So there also we can see that there is a high prevalence of heart failure conditions in these patients who are at maybe there's a correlation between both the kidney injury and the heart failure patients. So maybe if we come up with some AKI biomarkers that may help in early diagnosis and may help progressing patients towards the heart failure third stage. So these are few studies which are already known. So novel biomarkers which we have taken under our study would be involving the NGAL, cystatin C, KIM1 and NephroCheck along with other biomarkers of heart failure like BNP, SS2 and other like inflammatory markers like IL-17, IL-2 and all would be there. So it has been seen that NGAL and cystatin seems to be promising, but not yet routine clinical practice being done. So we'll need for the validation of early renal biomarkers in PCI-treated STEMI patients. So the lacunae here is that the role of LPGs, LDLs and ApoBs are already studied. However, the role of inflammatory and AKI biomarkers remains there. So therefore, we'll be studying both of them, where the objectives would be we'll be screening the inflammatory markers along with AKI biomarkers. Then we'll demonstrate the correlations between biomarker profiles and clinical variables, and then identify signaling pathways linked with STEMI and HF pathogenesis. So these are few data points which we have come up, where we had put up the real-time PCR of cystatin, NGAL, and KIM1, and we found that yes, they found to be peak. The levels were found to be at peak at both the the shorter time points like 6 hours and 12 hours. However, if we look at the long-term time points like 48 hours or 72 hours, or maybe at the time of discharge, if we see, so both KIM1 and cystatin, they were found to be rising early and they were acting as early markers of AKI and cardiorenal risk. However, there was a decline at the discharge, which may suggest that there is a transient kidney injury, but persistently high levels were there at discharge, thereby predicting the worst prognosis. So similarly, we checked for few inflammatory markers like IL-17, where we found that it was found to be maximally expressed in the reduced ejection fraction, followed by mildly reduced and the preserved. Further checking the levels of TNF alpha, IL-1 beta, and GDF. So again, we found that they were rising more and gradually, peaking at 24 hours and remains elevated longer. So this is how, if we find that yes, even the levels are there even at the time of discharge, that may predict towards the worst prognosis. So now, why there is a need for this lipid guideline for our country? Are we different? So there comes the role of atherosclerosis. So there are few risk factors which are there like diabetes, hypertension, tobacco, and the family history, which define what is known as dyslipidemia. So if I talk about why there's dyslipidemia in the young, why we can see why the prevalence of being seen higher in younger patients. So this is maybe because of other risk factors like LDL cholesterol, HDL cholesterol, or maybe CRP and other genetic genes which are already known. So what we had done, we had run the metabolomics of few patient samples. So here what we did, we characterized both HF and STEMI patients in terms of LDL, HDL and CRP, and then we found which all patients come under the category of atherosclerosis. So we ran the metabolomic data for these patients, and there we found few compounds which were found to be significantly expressed in few of the patients. As you can see that we had taken different time points of MI at the time of discharge, 48 hours, 12 hours, 24 hours, so as to maintain the uniformity in the data. So there we found that few acids like propanoic acid or maybe ethyl like glycol or lactic acid along with few glucose intermediates were found to be expressed significantly in these patients. And here we had taken ribitol as one of our standard marker. So the take-home message here was that the CVD events is there and very young adults are increasing. So diabetes and lifestyle risk factors fuel the early CVD. So these novel biomarkers, if we can dig in using metabolomics or proteomics, they may offer promise in early detection. So this is another like little piece where we are as we are collecting these HF patients, we are also checking into the fatty liver patients, how we can characterize this CVD and fatty liver together, where we have come up with PNPLA3 as one of a protective factor. So what we have done here is, as PNPLA3 gene is seems to be implicated in fatty liver and metabolic diseases, where G allele is linked to fatty liver progression. So there we are categorizing our patients into CVD alone, CVD and fatty liver, and fatty liver alone. So here our objective is to evaluate the protective role of PNPLA3 in different patient subsets. So what we have done, we have taken these patients, we have taken DNA isolate, DNA has been isolated. If I look at the methodology overview, so our post DNA extraction RFLP analysis has been done, and we have looked out for the CC, CG or GG genotypes. If we can look into the allele and genotype frequency calculations, then it would be helpful for us to correlate them with the clinical parameters. So these are few patient samples which we have run up. As you can see, some have been labeled as HF and fatty liver, few are only HF, that is only CVD, and others, like the others where we found out was that these are few genes where we have found the mutant type bands for this PNPLA3. However, the others show the wild type pattern. So after calculating and putting the genotype and allele frequency together, we could find that CC was little, the one of the recessive genotype was there in only one sample. In if I talk about 150 samples we collected, so it was there only in one of them. However, GG and CG genotypes could still be seen. So this is how we have thought of that. We are trying taking more number of samples right now so that it can be of some publishable data. So we'll look into these clinical parameters of all these genotype and allele frequency phenotypes which we have got as of now, and then we'll look at the biomarker potential for early identification of high risk patients out here. So this could influence the personalized therapy approaches targeting the lipid metabolism. So lastly, I would like to talk about the last project which we are working on is the gut microbiome and heart failure reduced ejection fraction patients. So here we'll be taking these HFR-EF patients. So we'll be running the 16S rRNA sequencing out here using NGS, and then we look out for few markers like TMAO, which is already known to be one of a major factor leading to CVD or maybe leading towards the progression of heart failure. So we look at the levels of TMAO along with some short chain fatty acids. Then we'll look out for altered bile acids, amino acids, and inflammatory metabolites. Specific species which we'll be targeting is the Collinsella aerophaciens, because this is seems to be, it's already reported in literature that it seems to be one of a major bacterial species which is seen to be prevalent in the cardiac patients. So taking into account all the metabolites and the specific bacterial species, if we can think of, then we may try correlating them with the endothelial dysfunction, vascular inflammation. This may help in predicting towards whether it is worsening the HF patient condition or not. So thereby the clinical translation of this part would be these biomarkers may help in diagnosis, phenotyping and prognosis. We'll also go ahead with the FMT, which may help restoring the healthy microbiome. And few precision therapies would be there which would act as either in terms of probiotics or antibiotics. So that is it. Thank you.